Good morning, folks. This video is on translation in eukaryotes. Let me share my screens. I have two screens today, and here they are. So what we'll do is we'll follow the PowerPoint on the left screen, and then I'll make some drawings and annotations using the tools on the right screen. And as you know, I am a very, very strong advocate for using graphical tools when appropriate. All right, so what do we say with this slide? This slide simply says that when you look inside the cells of a eukaryote and prokaryote with respect to translation, prokaryotes, they have a single type of RNA polymerase. And that RNA polymerase does the transcription for all the genes that prokaryotes have, right? So remember, polymerases are enzymes that put together nucleotides. RNA puts together RNA nucleotides, and DNA polymerase puts together DNA nucleotides. So they have one, and we, eukaryotes, have three. And why do we have three? Because there's a division of labor, right? So some of us, some of our RNA polymerases, for instance, number one, RNA polymerase 1, as you can see in the chart over here, it's been sequestered to dedicate itself to copying the ribosomal RNA genes. So type 1 is concerned within the nucleolus. Remember the nucleolus from chapter 1 is a darkly staining region of the nucleus, where there's a separate biochemical activity going on. What is that biochemical activity? That's the formation of new ribosome subunits, their proteins, and their RNAs. So here, ribosomal RNA is being transcribed, right? And once the RNA is made, it'll be incorporated with proteins into the small or the large subunit of the ribosome. Then we have type 3. Type 3, RNA polymerase, is responsible mainly, as you can see here, for tRNA genes, transfer RNA genes, but it also helps with a small subpool of ribosomal RNA genes. But more and more evident, more recently, is that it's also involved with small nuclear RNAs, which are important in regulating the behavior of genes, and many other types of newly discovered RNA species, right? So that's going to be normally involved with tRNA and other types of regulatory RNAs. So let's put that, tRNAs plus other types of RNAs. Okay, so type 2, the one that's left over, is mainly going to be involved with copying what we classically call genes that produce proteins. So that's going to be messenger RNA that then goes down outside the nucleus to the ribosome. And at the ribosome, it's then converted into polypeptides, right? But there's also a plus sign there. And here we have siRNA, microRNA, and then you can see on the far side, we have small nuclear RNA, et cetera, et cetera, et cetera. So, you know, there's a general distinction between the three types. We have so much more genetic material. We have so many more genes that nature has decided to divide the workload amongst three populations of RNA polymerases. So the next thing that we need to know, I'm just moving down the PowerPoint that's available to you guys on the CAT course website. The next thing that we need to know is the way that this one here works. So we're going to focus in on RNA polymerase 2. Once you understand that, the others operate in a similar fashion. So let's move down to the next slide and use that space, right? So what's the important learning experience here? Well, we need to know the order in which these components come together before you even get transcription beginning, right? So the order of activation. And unfortunately for us, this is a bit more complicated than than it is in prokaryotes, right? So the order of activation is more complex. So what do we mean by that? Well, let's keep everything general for the time being. We do know that transcription has 3 stages. The first stage is initiation. The second stage is elongation. And the third stage is termination. What does that mean in English? That means when a particular gene is transcribed, whoops, I'm not that good with them. Oh my God, that's terrible, but you get the idea. That's the gene, and that gene will be activated at a switch, and the switch for that gene will be sitting somewhere here, right? So that's going to be a region of DNA, which we call the promoter. Now, promoters appear to be more complex in eukaryotes than in prokaryotes. So we may have elements here, DNA sequences, that may help or hinder the activation of transcription from the beginning of the gene, right? The important thing is the entity that wants to bind to the beginning in prokaryotes is the RNA polymerase. The entities that bind initially to the promoter in eukaryotes are other proteins. So other proteins will come and land in this region here, and those proteins are brought in a particular order. Other proteins will land over here, and maybe some further down, and then, only then, will RNA polymerase be sequestered into that complex. And that's what we see happening on the PowerPoint on the left-hand side. So there's your Tata box. That's just the part of the promoter which is necessary to be attached to. And the first thing that comes in is transcription factor 2D. Why is it called transcription factor? Because it's a protein that helps in transcription. The 2 stands for RNA polymerase 2, right? And the last letter is to distinguish it from other proteins that are involved. So we need to know the order of activation for initiation, right? So under initiation, we say that TF2D is necessary, but it has a partner called the Tata binding protein, right? So that you can see just there in dark green. So these 2 have to first combine, and then they come and sit down on the promoter right there. Then around them, other transcription factors will appear. So the next one is transcription factor 2, and that's going to be B as in Bravo. So that comes in next. You can see that in brown. And then a bunch of other things happen, including the arrival of transcription factor 2. Yes, I was going to say F, but E comes in first, right? And then transcription factor 2F first binds to the ribosome, not the ribosome. Did I say ribosome? Terrible. It binds to the RNA polymerase. And once it's bound to the RNA polymerase, then the 2 of them are brought in next. You can see that happening right there. Okay, and then transcription factor 2H is then brought in. Oh my God, I'm running out of space. So 2H also comes in, and other factors that we don't need to talk about. And only when that happens do you have the necessary machine building up part by part on the promoter for this particular eukaryotic gene. And once that happens, then other factors will decide how much of a push or how much of a hindrance there will be to the movement of the RNA polymerase 2 down into the gene so it can start producing some RNA. So that is initiation. Initiation is a sequence of activity that we need to learn. And this slide here simply gives you a great insight into how that works. Okay. So here we see a picture of one of these complexes, and you can see the tail of the RNA polymerase 2. The tail is going to be very important in the next section. And the tail is used for bringing in other protein complexes that are going to help process the pre-messenger RNA, the RNA that's coming out from the back end of the polymerase. So initiation is at the top. And then elongation, this step here, you can see taking place now at the bottom panel of this slide. You can see some of these proteins have done their job and they've been now removed to be used elsewhere. But some proteins are still there, transcription factor 2D with the tartar binding protein. And you can see other proteins are now climbing onto and attaching to the tail of the RNA polymerase 2. And those proteins will interact with the newly made RNA. And as the RNA is made inside the heart of the enzyme, the tail will get longer and longer. And as it passes by this protein complex, something will happen to the tail. As it passes by the next protein complex, something else can happen to the tail, the RNA, right? So let's have a look at that. This next image was taken from another book, just to show you how this pre-initiation complex builds up over here. So this is from a more detailed biochemistry cell biology textbook. But if you look carefully, it's got all the same parts coming together as we had earlier. The only difference is the angle at which the DNA, presented here in orange, is bent by the transcription factor 2D, right? So transcription factor 2D has this profound effect of twisting and turning the DNA into a hairpin. Most students think that DNA is rigid. It's not rigid. It's very flexible, like a piece of hair, right? It can be bent and twisted. So this is very important to access the sequence of the promoter, the tartar box. So if you look at that in a clear light later on, you'll see that will make sense, right? One thing that we do recognize now is that there's so many genes in eukaryotes, up and down all the different kingdoms, that there are many different plans that have evolved. So this slide is simply telling us, well, certain genes have this pattern, other genes have this pattern, and yet other genes have a different pattern in terms of their activation, their initiation, right? In some cases, just a few, we found DNA sequences downstream of the start point, i.e. maybe within the gene itself, that are going to influence the activation of the gene. In most cases, the sequences are upstream from the start site. So this arrow represents the very first nucleotide that is part of the RNA, the very first nucleotide at the beginning of the RNA molecule, right? So that's what we call the start site. Regardless, you can see the sequence can vary between different recognized categories of these structures. And the proteins that bind to them, they have preferences for which sequence they are seeking in order to sit down on the DNA. This is my favorite picture from this chapter because it reinforces the principle that DNA sequences a long way away from the promoter itself, from this region here, a long way away, also have a say in this committee decision whether to start transcription or not, or by how much, right? So this protein here has sat down on this special sequence of DNA, which we call the enhancer, and it binds to the enhancer, and that protein will then help increase the rate of transcription of this gene that you see in red by bending the DNA around and then attaching to a giant protein called the adapter or the mediator. And then that will influence the shape of this protein, and that will have a knock-on effect on the shapes of other proteins, and therefore influence how much or how little the RNA polymerase will be allowed to move forward in its elongation phase. So that is a committee decision, right? Should this protein be missing, maybe from the diet or maybe from the cell, then that interaction won't take place, and maybe the polymerase will be that much less likely to move forward or to produce less RNA from this particular gene. So it's like a dimmer switch, right? Where the activity of transcription, the rate of transcription can be increased or decreased depending on which combination of proteins are present in that vicinity of DNA. So this is the main difference between prokaryotes and eukaryotes. Prokaryotes, pretty simple. One, maybe two factors decide, and they have a quick decision, a small committee. Whereas with us, it's a giant committee with many seats around the table. Okay. This section is not important about DNA tension, so we'll just bypass it. This slide is the next most relevant slide. What it's saying is that when you compare prokaryotes with eukaryotes, which is the ultimate goal of this chapter, what you have to do is keep an eye out on complexity, the overall complexity. What the top half of the slide is relaying is the situation with prokaryotes. Now, what it's saying is we find one of two patterns, right? Let's call them I1 and I2. Okay, so the first pattern is you have a gene. The gene starts here and it goes down to about here. That gene will have a switch at the beginning called the promoter. It may have other parts like the operator and the enhancer, maybe. It doesn't matter. So that's the switch, right? You bind to the switch, simple decision, and then you transcribe into the gene itself. So they have a very simple architecture. Sometimes they go one step further. And what they have here is a single switch like that. That's your switch with the promoter, the operator, and other stuff. And then they are able to code for a couple of different polypeptides, all from the same switch. So this thing here will produce a single, long, multi-dimensional RNA molecule. And that RNA molecule will then be coded just for that region to produce this polypeptide. And then another polymerase, sorry, wrong word, another ribosome will bind there and produce a different polypeptide. And then another ribosome can bind there later on to produce a different polypeptide. So in prokaryotes, they have things called operons. Operons. Operons, in my mind, are simple. They are complex genetic organization where you have one switch plus multiple genes, right? Yeah, and all three genes in this case are controlled by the same switch. In this above example here, we have a switch plus one gene. What do we find in eukaryotes? Well, so far we've only found operons in very, very specific species of yeast. But for most organisms, there are no such thing as operons, right? So we do not find operons in general in eukaryotes. Do we find single genes with their individual switches? That is the most common situation we find, right? So in our case, we have a single switch. That switch is much more complex than the switch that you have in prokaryotes. And that switch is then attached to a single gene. And the gene is then transcribed along the DNA, right? So the gene is here and the switch is here, right? And in our cases, we can have parts of the switch that are way over there or even a long, long distance away over here, right? And those DNA sequences will bind their own proteins, and those proteins will then somehow influence the activation of this gene from that switch. So this is what we have compared to them. And this is what this is saying, right? Now, the bottom part of this simply says, hang on a second, the product of that gene, yes. So this is the next level of complexity. What it's saying is that the RNA produced from here, from that gene, is going to be further modified by three mechanisms that are common. A is a cap is put on. We're going to look at that in a few seconds. A cap is always put on this side of the molecule, right? That end. What end is that? That's going to be the 5' end of the messenger, or the RNA, to be precise, right? Then B, the end of the molecule is tailed. So a tail of special nucleotides, we call that tailing. A bunch of special nucleotides will be put on somewhere along this length here, normally a bunch of A's. How many? That can vary from a few dozen to hundreds, several hundred. Okay, so that's called tailing. And then there's a last one called C, called splicing. So for many genes, not all genes, for many genes, segments of the messenger RNA are removed and the remaining segments are brought together, just like somebody cutting film in a director's booth. Oh, this scene is not necessary, let's cut it out of the film, right? So these regions called introns are cut out, and then the remaining parts called exons are spliced together. That seems to be very common in eukaryotes and not in prokaryotes. So these are the main differences. So the machinery to do all this A, B, and C is more complex than you find in anywhere else. So we have to learn that. So where do we begin? Well, let's start by just focusing on this image here. So hopefully by my commentary a few minutes back, you know that the nucleolus, which is inside the nucleus, normally a dark patch, could be more than one, but normally one dark patch inside eukaryotic nuclei. That nucleolus is a hotspot of activity to do with ribosome subunit construction, right? Genes and proteins are being heavily incorporated into the subunits of ribosomes. Then the ribosomes are passed out into the cytosol through the nuclear pores, and that's where they hang around until they're needed to produce polypeptides. So polypeptides are not made inside the nucleus. Polypeptides are made out there in the cytosol. So we mentioned the tail of the RNA polymerase. So let's move our attention down to that. So the RNA polymerase, the enzyme that copies DNA into RNA, it has a tail, right? And that tail is simply part of its polypeptide subunits. That tail has enzymes that sit along it. As you can see on the other side, we have a different combination of enzymes. That's what triangle, square, and a circle, right? So the green in this case are this one here, the triangle. Those are considered to be capping enzymes, right? They add cap, the 5' cap that we talked about earlier. They add the cap immediately. Once the tail is long enough, they add the cap. That makes sense, right? So that's easy to do. So as soon as the messenger RNA begins to emerge, or the pre-messenger RNA begins to emerge from within the polymerase, gets long enough, the 5' end of that molecule is capped. The cap is put on to protect it from harsh environments in the cytosol. Then another bunch of enzymes, they do the splicing. So when a splice site comes along next, this is an intron, this complex will recognize that, and cut this piece out, and then stick that piece onto that piece. So that makes sense, right? Everything's done in place. And then we have the blue factors. And they wait until the last part of the pre-messenger RNA is made, and then this complex here will do its magic by putting a poly A tail onto the end of the pre-messenger RNA. So a very elegant solution by Mother Nature to achieve that result. So that's something that we need to understand. So the end product is a pre-messenger RNA that has a cap, then it has a tail added to it, and then before it's finished into messenger RNA, the introns are removed as well. Now we have the finished messenger RNA, the finished product, right? And that finished product is then sent out into the cytosol where the ribosomes are, and the ribosomes will grab one of these and start converting it into a polypeptide, and then the polypeptide will eventually become a functional protein inside the cell. So you can see that, okay? What is the cap? The cap is simply a backward guanosine with a methyl group added to it. It's a nucleotide, but it's strong enough to protect this end of the messenger RNA from attack in the cytosol by enzymes that like to attack a messenger RNA. And you can see a comparison of the size of genes, right? So again, a very simple architecture for prokaryotes and a more complicated architecture for eukaryotes. So that image is very important. And this slide simply says, oh, some of our genes are tiny. They have a few introns. Yep. Some of them are huge, as you can see, factor 8, a blood clotting protein that can be 200,000 base pairs of DNA from one end to the other. So there's no reason or rhyme as to the size of a gene. It's based on evolutionary needs. Okay. The last thing we need to tackle right now is how do you perform the removal of introns? Well, when we study enough introns in this picture are going to be in orange, and the exons are going to be in blue. So this is just a prototypical intron, any intron. So we have a consensus here, right? The most important thing is that this side is recognized by a bunch of proteins, the internal side of the intron, the 5' end, and the 3' end of the intron is also recognized by proteins. So the proteins must be able to distinguish this piece of DNA from neighboring pieces of DNA, right? So the sequence here is very important. And there happens to be a small region a bit further in from the 3' end of the intron. That's also important, where that base A is very important. So the machinery that we're going to talk about in the next few seconds is going to grab this, grab that, and find that, and then remove this intron. That's the way it's done. We'll come back to this in a second. So let's move to the next screen. And here you can see on this screen how the lariat structure, like a cowboy's lasso, how that forms during the removal of these introns. So this is just nature's solution on how to handle precisely the removal of introns down to the precise base. Any errors will lead to the polypeptide not being produced correctly at the next phase of translation, right? So something has to grab this end of the yellow region, something has to grab that end of the yellow region, and something then has to cut and then move the DNA between these 3 points. You can see the first cut is made right here at the 5' end of the intron. And then that 5' end is moved onto the A. The 3' end of the intron, no, the exon, the 3' end of the exon is held onto so it doesn't drift away. And then it is precisely brought in close proximity to the other exon over on this side. And that exon are then combined together to produce a perfect cut and paste. And then the lariat, this structure here, is released into the nucleoplasm, the nucleus space of the nucleus. And it can either be digested back down into nucleotides to make fresh RNA, or in special cases, some of these regions are used for something else, right? That we don't need to get into today. When we look at different genes, we find that there are more proteins than there are genes inside your body. So we have about 25,000 protein coding genes in our DNA, but we have over 100,000 different types of protein or polypeptide. So how does that make sense? There's not enough genes to make this much protein in a human being. So what we've discovered is that the introns can be removed in different tissue using different patterns, right? So striated muscle is muscle that forms your biceps and other skeletal muscle that you can move at will. And there, the pattern of splicing out these introns is of the type illustrated in the top row. Then when we look at smooth muscles can be found lining your blood vessels, for instance. So most of these are non-voluntary muscles that take care of themselves through an alternative system. So they cut their messenger or the pre-messenger RNA, they cut them into this pattern to form the final messenger RNA. So in those cells, this is the pattern that's produced. In skeletal muscle cells, this is the pattern that's produced. Further research has revealed that in fibroblasts, which are present in connective tissue, they use an alternative pattern. And indeed in brain, that uses a completely different pattern. So a single gene inherited from your parents, right, as duplicate copies, that single gene along your DNA can be spliced into at least, in this case, 5 alternative polypeptides. So therefore, that explains why we need such few genes to produce such fast alternative versions of the polypeptides. So alternative splicing has a role. It's not just a waste of energy in making those introns. Those introns have some benefit that we gain from having them. Okay, so that is all we need to know about the mechanisms that operate in eukaryotic transcription. The next video will show you how the messenger RNA is converted into polypeptides. I want to thank you, and if you have any questions, office hours are the best place to come. Thank you so much.